Bismillahirrahmanirrahim. Today I would like to share the knowledge about toxoplasmosis. That one is the most important and critical disease. My name is Sarfraz Rahman. I am a PhD scholar in the Department of Parasitology. I would like to share this knowledge with references these books, the Veterinary Parasitology, second edition by G.M. Arkohaj, Veterinary Parasitology, fourth edition by M. A. Taylor, George's Parasitology for Veterinarians, that 10th edition by D. White, D. Bowman, and a handbook of veterinary parasitology, that the domestic animals of North America by Henry J. Griffiths, taxonomy of Toxoplasma by Norman D. Lowine. That one is the research paper. A veterinary parasitology reference manual, 5th edition by William J. Fourier, and last one is the internet sources. You can see these two pictures, that is the reflect or indication of that this disease toxoplasmosis, and this one is the pregnant women, and this one is the cat, and these both have the relation to induce this disease. And similarly, this picture is also indication of this disease. And please subscribe my channel, and also push the button of bell icon so the new video will come to you as soon. So the subphylum or the sporozoa or Epicomplexa, this sporozoa or Epicomplexa, it is divided into the main three classes. The first one is Coccidia. It is also called the elementary sporozoa. The second one is the Pyroplasmidia, and third one is Hemosporidium or blood sporozoa. So the first class that is divided into the two main classes. First one is the Emerridae, and second one is Sarcocystidae. So this family Emerridae, that is divided into the following genera. That first one is Emeria, second one is Isospora, and third one is Cryptosporidium. The second family, that is named Sarcocystidae, and it has the following genera. That first one is the Toxoplasma, Neospora, and then Hemondia and Basnoita. So that this Toxoplasma, today I would like to share this knowledge about this genera, that is the Toxoplasma. And second class, that is the Pyroplasmidia, that you have the following genera. That the first one is the Chileria, Babesia, Cytoxazone. And third class, that is Hemosporidia, or other name is the blood sporozoa, and it has the following genera. That the Hemoproteus, Plasmodium, and Leucocytozone. So the sporozoa are Epicomplexa. What is meant by Epicomplexa? The group has possession of any apical complex. And what is the apical complex? It is the structure which apparently assists penetration of the host, and it is visible only with electron microscope. And you can see this picture. In this picture you can see that this structure is like the apical, and it is helpful to penetration. And having apical stages in their life cycle, trophozoites have no cilia and no flagella. These are the intracellular parasites. And the sexual stages in their life cycle, that is called the gametogony, while the stage at which the gametes produce spores is called sporogony, and while another that the asexual stages, that is called the schizogony. And it is divided into the three main classes, that the coccidia or elementary sporozoa, pyroplasmidia, and hemosporidia or blood sporozoa. So that this family Imiridae, and these organisms are intracellular parasites, and schizogony and gametogony that take place into the same host, while the sporogony or maturation takes place outside of the host. So that it has the three main genera, that is the Emeria, Isospora, Cryptosporidium. So the coccidiosis. What is the coccidiosis? It is the infection of Emeria or Isospora, or collectively of the both genera, that the Emeria and Isospora, that is called the coccidiosis. And these structures, you can see that these are the Emeria ovocyst, and you can see that it has the four sporocysts. These are the four sporocysts, and you can see in this picture. The important points, the coccidia have the following five genera, and you can and create the difference between these five genera on the basis of the ovocyst structures. And the Emeria, it has the four sporocysts and two sporocytes. Isospora, it has the two sporocysts and four sporocytes, while the Cryptosporidium, that have the two sporocysts and four sporocytes, while the Toxoplasma, and it has the two sporocysts and four sporocytes. So today I would like to share the knowledge about this Toxoplasma. So keep in mind that the Toxoplasma have the two sporocysts and four sporocytes, while the Sarcocystis, it has only the one sporocyst and four sporocytes. So the toxoplasma that causes the disease, and its name is the Toxoplasmosis. It is actually the disease of blood and lymphatic systems, and it is also a zoonotic disease. And you can see this picture, that is Toxoplasma, that first it adherent to the cell, then it enter inside the cell, and in VN and intracellular application, then it is excluded from the outside of the cell. So the first description, the Toxoplasma gondii, that species, firstly it was described by the Nicoli and Manciaux in 1908, and the 7 species are recognized. Number 1 is the Toxoplasma gondii, and it was discovered by the Nicoli and Manciaux. The type of the species from the about 200 species of the mammals and birds with the oocyst in the phalanx. Number 2 is the Toxoplasma Elencheri, that is discovered by the Da Costa and Pirari, and it is discovered from the frog, that is the Leptodactylor oculatus, and Toxoplasma brumpti, that is discovered by Court Line from the iguana that tuberculata. And fourth one is the Toxoplasma colibri, and that is discovered by Thibault, and from the snakes, that is the Colibur maliricus, and Colibur that is the Iridiflus. Next one is the Toxoplasma himondi that is discovered by the Frankel Dubé, and a new combination for himondi, AO himondi, and from the house mouse with the overcyst in the domestic cat. And next one is the Toxoplasma reni that is discovered by the Levine and Nye from the leopard frog named the Rana pipiens. And the last one is the Toxoplasma serpi that is discovered by the Scorza, Doggett, and Itorisi erocha from the toad that is formal in your this. So the toxoplasmosis in humans and different animals. The toxoplasmosis in humans, sheep, goats, cattle, dogs, cats, birds, reptiles, amphibians, and mammals. So the toxoplasmosis in humans, the main source or main species, that is the Toxoplasma gondii, and definite or the final host, that the all felids or domestic cats, intermediate host, that the human being or birds, all mammals, sometimes cat, and the habitat inside the human being or reside in epithelial cells of the intestine, then it moves to in the blood. And the sporulated oocyst, that is identified by the microscopy, and 12 to 10 micrometer oocyst, that the size, and 4 to 17 micrometer Chazon size, or 6 to 8 micrometer Tachyzoid size, 100 micrometer bradyzoid size, and 10 micrometer size of the gamete, and 1 to 5 days the oocyst, the sporulation take place, and 1 to 2 weeks shedding of the oocyst, it needs this time period. And the several thousand organisms in the bradyzoids, and 8 to 16 organisms in tachyzoids, and 32 merozoids in the chazons. So you can see that is the congenital transmission, and you can see the head of this baby, that is actually the infection, or it is the toxoplasmosis. So that one is the shape of the tracheozytes, you can observe in this diagram. So the Toxoplasma gondii, it is belong to Epicomplexa, and it is a coccidia or heteronexus organism, obligate intracellular parasite, primarily infect the intestine and intestinal coccidia, or it is also the zoonotic parasite. And you can observe this Toxoplasma gondii shape of this. You can observe that conoid or other structures in this diagram. So that the strains of the Toxoplasma gondii has unusual clonal population structure consisting of the three widespread lineages known as the type 1st, 2nd, and 3rd. Type 1st is highly virulent. Type 2nd and 3rd are relatively non-virulent. In the type 1st, that the GTI strain that is present in the goat, and RH strain that is present in the human, and it's that ATCC50838. And this is similarly that the this strain is present in the sheep, and type 2nd, that the B7 strain that is present in the sheep, and ME49 strain that is present in the human, and DEG, that strain present in the human, and type 3rd, that the CTG strain that is present in the cat, and VEG strain that is present in the human. The subtype of microorganism is called the strain. So you can observe this in picture, that belonging to type 1st strain, this belonging to type 2nd strain, or this actually belonging to the this type 3rd strain. So the surface antigens of the Toxoplasma gondii has the following surface antigens. SAG1, it has the 319 amino acids, its protein is P30, and SRS1 that have the 420 amino acid, SRS2 that 372 amino acid, SRS3 that 345 amino acid, and its protein is P35, and SRS4 that 396 amino acid, and SRS5 actually it is unknown the amino acid, and SRS6 that 393 amino acid, SRS7 no absorb this the amino acid, and SRS8 similarly no yet discovered the amino acid, and BSR4 that 398 amino acid, and it is also called the P36, and SAG2A that 186 amino acid, and P22, and SAG2B that is 190 amino acid, SAG2C that is 365 amino acid, and SAG2D that is the 373 amino acid, SAG2E that 340 amino acid, SAG3 that 385 amino acid, and it is also called the P43, and SAG4 that 172 amino acid, and it is also called the P18. SAG4B that 180 amino acid, and similarly you can see that the SAG that the 5A 362 amino acid, and SAG5B 367 amino acid, and SAG5C and 367 amino acid. So these are the surface antigens of the toxoplasma, and these surface antigens are actually helpful for the diagnosis of the infection of Toxoplasma gondii, and these are the very important, and you can see the structure of this that the surface that the tachyzoids, and the upper stage is the conoid and micronemes, Roptres protein, Epicoplast and Golgi body, nucleus, endoplasmic reticulum, dense granules, and that is the mitochondria. It is also present in this structure. So the prevalence of the toxoplasmosis, 84% in the France, in especially pregnant women, and 32% in New York in pregnant women, and 22% in London in pregnant women, and similarly the 16 to 40% the population is infected in United Kingdom. 50 to 80% population is infected in Central and South America and subcontinental and continental Europe, and overall prevalence in Pakistan in human, that is the 48%. In cattle, that is 19%, and goats 25.4%, in sheep 19%, in cat that is the 26%. These values are actually studied from the research papers. So the transmission oral route, it's a very important that if you drink the cow's milk that is infected with the toxoplasma, and similarly unboiled goat's milk you can drink and you are actually getting the infection, and raw hen's egg it is also source of the infection. But the sexual activity like the kissing, it is not important for the transmission. And it is very important that eating of the undercooked or raw meat, that is the main source of that spreading the infection. And blood transfusion and organ transplantation, and then the horizontal transmission, vertical transmission, that is the transplacental transmission, it is also called the congenital transmission. You can see that this is the source of the placental transmission. Tracheostomes are moving out and actually causing the infection in that the newly baby. And the other, you can observe this that the cat is the main source that is spreading the infection in the human being. And similarly in this picture you can observe this one. So pathogenesis, first of all, oral ingestion, Tracheostomes in viral form, and it is spread in the blood, and then disseminates through the whole body, and actually infection and in nucleated cells and multiplication take place, and cell destructions and necrotic foci, and it moves to the, and it causes the surrounding inflammation, and it causes the tissue cyst, and these tissue cysts actually cause the big source of lifelong chronic infection and onset of the CMI. So you can observe in this structure, and that the whole cell lysis and 60 hours and post infection, and then it spread this infection, then goes inside the cell, and then multiplication, then spread the infection, and that is the source of this that the toxoplasmosis. So clinical signs, you can observe it's a very dangerous disease. Symptom of the toxoplasmosis, fever, lymphadenopathy, that you can observe in this picture, that is the lymphadenopathy, and you can also observe in this picture. And the stillbirth, this the picture actually showing the image, and a child with severe brain damage or vision problems, abortion, and chorioretinitis. In this picture you can observe that the chorioretinitis and hydrocephaly, and this picture is actually the hydrocephaly, and it causes the chorioretinitis also. And microcephaly, you can observe in this picture that has skull that is very small, and headaches, malagia, stiff neck, anorexia, sore throat, arthralgia, and rash. Or even that you can observe that the ARHs and eye pain, abdominal pain, and nausea. So these are the main clinical signs of that toxoplasmosis. And diagnosis, you can diagnose with at direct microscopy through the feces and smear and section stained with ginseng stain. You can observe that in this slide, that the tracheostomes and this structure of that oocyst of the Toxoplasma gondii. Another method of the diagnosis like the PCR and Sabin-Feldman dye test, that is the gold standard test, and ELISA and IFA and latex agglutination tests, that you can identify this disease. Treatment, it's very important that the pyrimethamine, doroprem, that 1 milligram per kg body weight orally for first 3 days, followed by 0.4 milligram per kg body weight, and its mechanism of action is actually that the inhibit the dihydrofolate reductase, that cause reducing the production of folic acid, and this folic acid which is required for the nucleic acid synthesis. Next one is the sulfonamides. It's include the sulfadiazine, sulfadimazine, and sulfamethoxazine. Its dose rate is 100 milligram per kg body weight orally once and may be increased up to 4 gram orally divided doses. And the commercial name of this disease, that the medicine, that is the Spectra, and that it has the trimethoprim and sulfamethoxazole, 80 milligram of TMP and 400 milligram of sulfamethoxazole, one tablet for every 12 hours for 14 days. And spiromycin, that 2 to 3 milligram per day divided into the dose in men, and azithromycin 2 to 4 gram per day in men, and clindamycin 150 to 300 milligram per adult man and dog. And monensin, that is important, and this medicine actually the dose rate is 5 milligram per kg body weight in sheep. It modify and actually its mode of action, that is it modify the movement of ion across the biological membrane, and it helpful in the stop of the abortion in sheep. And SDD, that diamino diphenyl sulfone, that had dose rate is 100 milligram per kg body weight for 14 days, is the most effective treatment, and its mode of infection is that inhibit the bacterial synthesis of dihydrofolic acid, and it actually inhibiting the nucleic acid synthesis. And next one is the sparomycin. It is actually the drug of choice for this disease. It's actually prevent the vertical transmission. It is a microreal antibodies, and that cannot cross the placenta, and it remains concentrated it, and its dose rate is 1 gram orally every 8 hours for the duration of the pregnancy. And pyrimethamine or sulfadiazine in case of the confirmed fetal infection, and it's actually targeting the infective stage of the parasite, and it is current gold standard for the treatment of this toxoplasmosis. And decoquinate, that it actually that to stop the abortion in mid gestation, and it is also important, and you can observe in this picture that is the tablet of that the spermicin.